Welcome to South Bend News. Today we have a report on stem cells. The use of embryonic stem cells has long been a topic of debate and controversy in the scientific community. However, a new study from Drs. Takahashi and Yamanaka of Japan indicates an alternative to the use of embryonic stem cells for research. Let's turn to our field reporter, Jeremy, for some background on this fascinating revelation.
Thanks, Kevin. Well, everyone, I'm here at the Notre Dame Duncan Student Center with the basketball team's newest recruit, Biggie Hoops. Biggie here is going to help me explain the concept of stem cells. You see, about a year ago, Biggie was tearing it up out there on his high school basketball court. He was hitting triple doubles and winning state championships. And college coaches all around the country had their eyes on him. He could have gone to any university he wanted. Now imagine Biggie is a tiny, microscopic stem cell. Right now, he's what the biological world calls a pluripotent cell. He can go to any school in the country that he wants, just like how a pluripotent cell can turn into any cell in the human body. Now, let's say Biggie here gets offers from 10 schools. He starts talking to the coaches, and soon he's committed to picking one of these 10. He has now become multipotent. He can still go to 10 schools or turn into 10 types of cells, but he's not considering any of the other hundreds of schools, and he can't turn into any of the other hundreds of cells. His options have become much more limited. A few more months go by, and now Biggie's narrowed it down to one school. He is satisfied with the offer and isn't going to apply anywhere else. He is now unipotent. He's one single type of cell and can't narrow down to any more cells. His decision has been made. But where did these pluripotent stem cells come from? Well, it all starts before birth. Let's imagine that this basketball here is Biggie as a human embryo, one of the first stages of human life in the womb. Right now, Biggie's made up of a group of cells forming the outer shell of the ball and a clump of cells resting on the inside. This combination of cells is what science calls a blastocyst. The cells resting on the inside of the basketball are the ones we want. Known by scientists as embryonic stem cells, these are the ones that can become Biggie and go to any school they want. They are pluripotent. They can be used to treat a number of diseases like macular degeneration, Crohn's disease, and blood cancers. Unfortunately, however, to get inside the basketball, you have to destroy it, which is okay for a ball, but for a human embryo, the goal is to preserve life. Haha. Anyway, it's a good thing these scientists are so smart, because they found a way around this problem. A scientist by the name of Shinya Yamanaka found a way to take regular human cells and manipulate them so they have the same qualities as the embryonic stem cells in the basketball. These new cells are called induced pluripotent stem cells. They can do anything the embryonic ones can, and best of all, you can get them without hurting any life. Now back to you, Kevin.
Thank you for that report. Now we have Dr. Yamanaka here with us today to talk a little bit about his research and what he has to say about stem cells. So Dr. Yamanaka, can you tell me a little bit more about stem cells?
Thank you for having me. So stem cells have two main characteristics. They have potency and they can self-replicate. Self-replication refers to stem cells' ability to infinitely create more of themselves by special kinds of division. And potency means that they have some ability to differentiate or turn into different types of cells. However, there are differing levels of potency. Pluripotent stem cells are stem cells with very high potency, as they can become any type of cell in the body, and any of the three germ layers, those being ectoderm, mesoderm, and endoderm. However, once pluripotent cells start to become more specialized, they undergo commitment and lose potency. For example, this can be seen as a pluripotent stem cell committing to become specifically an ectoderm stem cell, limiting the amount of cells it has the potential to become.
Where can we find these pluripotent stem cells?
Yeah, so pluripotent stem cells can only be naturally found in developing embryos. The blastula is an early stage in the embryo's development when it consists of a trophoblast layer that provides an outer layer of protection and an inner cell mass that contains the embryonic stem cells, which are pluripotent. However, the only way to isolate these stem cells is to destroy the blastula, which leads to major ethical concerns.
So how does your research relate to pluripotent stem cells?
So our research has allowed us to induce a pluripotent state in fully differentiated cells found in adults. So by inserting transgenes into a mouse fibroblast cell, we were able to ensure the expression of 4 specific factors or special proteins in the cells. And these were OCT34, SOX2, KLF4, and c-MYC. This combination of factors in cells allows for changes to take place to allow for the induction of pluripotency in a formerly unipotent cell. We were able to confirm the pluripotency of these cells by teratoma assays. So by injecting these induced pluripotent stem cells into mouse tails, we were able to observe teratomas, which is a tumor that contains all 3 germ layers. Because only pluripotent stem cells can produce all 3 germ layers, we were able to confirm the pluripotency. Additionally, these induced pluripotent stem cells, or iPS cells, are very similar to embryonic stem cells in morphology, which is their structure, and the expression of their genes.
That's great. I'm really looking forward to all the awesome work that has yet to come regarding your exceptional research. Thank you so much for coming on our show.
Yeah, of course. Thanks for having me.
That's all for today, folks. Thanks for watching.